so we'll be having conversation and things, but what we're going over today is G-protein, signal transduction pathway. And we'll be going over both the PLC G-protein pathway as well as the adenylate cyclase protein, G-protein pathway. After that, assuming that we still have time, we will move to the rhodopsin signal transduction pathway in the eye. So it's kind of similar, but different location and specific. Okay, so going ahead and starting off, this whole thing right here is the plasmolipid bilayer. This is a receptor, okay? So you have something bind to the receptor, and then that starts the G-protein pathway. The receptor then activates the G-protein. The G-protein is just a protein that's inside the cell, and it's kind of like a messenger that turns on other things. So, but it needs to be turned on first. So the receptor activates the G-protein to pick up a new, so right now it has a GDP. So the diphosphate, that's in the lower energy state, and so the G-protein's in resting state. As soon as the receptor turns on the G-protein, then it gets GTP to come in and kick off that GDP. So GDP is gone, it now has GTP, and GTP means that this G-protein now has energy, and it'll go over to PLC, which is phospholipase C, and the name tells you what it's going to do. It's going to cleave a lipid, a phospholipid, and we learned all that wonderful stuff in biochem about phospholipids. So phospholipase C will cleave a phospholipid. So PIP2, which is phosphoinositol, and I think it has 2 phosphates, is why it has a 2 there. That is what is cleaved. That's actually a lipid that's from inside the lipid membrane, and PLC pulls it out and cleaves it into 2 different things, okay? And where does it get the energy to do that? GTP. Yes, from the GTP and the G-protein going over and saying, do stuff, okay? So we'll go ahead and say this is kind of all step 1 over here. This is kind of the second little area over here. And then when this cuts PIP2, 2 molecules happen, because if you cut something in half, you're going to have 2 halves. Both of those halves are active, okay? You have DAG, diacylglyceride, I believe is what the G stands for. It might be glycerol. And that goes and activates protein kinase C. And by the way, this is really handy for me. If you remember PLC, goes with protein kinase C. That's always true, and it's really handy, okay? So diacylglyceride will go and activate protein kinase C. Protein kinase C is going to go ahead and phosphorylate stuff because it's a kinase, and what do kinases do? They mess with phosphates. Is this making sense so far? Yeah? Yeah? Okay. So it'll go ahead and phosphorylate stuff. And I'm just going to put an X here for any product, and then X with a P. It can also take phosphates off. Protein kinase does a lot of fun stuff. Okay, so that's what one of those things does. We now know that. What about this other thing coming off of PIP2? Well, it becomes IP3, inositol phosphate triphosphate, inositol triphosphate, IP3. IP3 goes down to the sarcoplasmic reticulum, and I'll go ahead and put SR right here, and it binds to a conveniently named receptor called IP3 receptor, and I remember that one because it's so nice, and it has the same name. Go figure. All right, so as soon as that binds, then what is stored in the sarcoplasmic reticulum? Calcium. So calcium comes out, and we're talking about smooth muscle as the example here, and so in this case, when calcium comes out, you have the calmodulin being activated because calcium is around, and that's what calmodulin does. It's modulated by calcium, calmodulin, and when that is turned on, it'll turn on MLCK, which is myosin light chain kinase, and it goes down and phosphorylates myosin. If you remember, myosin is a protein in the muscle. It's the one that has that head that kind of looks like that, and then it grabs onto the actin chain. And so in smooth muscle, MLCK goes down and talks to the myosin head to turn it on. Okay? So that is the PLC pathway. You start with the receptor being turned on, which activates the G protein. GDP kicked off by GTP. That gives it the energy to use the GTP, which right here, when the GTP is used again, it'll change back into GDP, and this will need to get another GTP if it's going to be activated again. Okay? So right here, the GTP gets used up. So I'll put GTP to GDP, because that's the energy being used to cleave this PIP2. Okay? Really quick, if you want to, try to do this first part in your head, just all the way up to PLC, and that way you can see if you got it. And then we'll do the next part. Just give a thumbs up when you're good. All right.
So,
go ahead.
PIP2, that's a phospholipid?
Yes. PIP2 is a lipid that comes out of the lipid membrane. I probably can draw it coming down like this. It just was kind of awkward. So it's being pulled out of here and being cleaved by PLC. So it's always floating around. PLC will break it when it needs to.
Okay.
So
it's a normal phospholipid?
It's a type of phospholipid in the lipid membrane, yeah.
Okay.
So phosphatidylinositol is phosphatidylinositol is what it's called. Why it's abbreviated PIP2, I haven't really been able to figure out. But it's also one of the ones our biochem professor wanted us to know. She used it as one of her examples in glycerophospholipids. So, yep, it's just one that gets cut, and then it's a signal in two things. So
It has a sugar on it.
Yeah, it's got a glycerol backbone.
Okay.
Like most
And so the
lipids.
DAG is the glycerol?
Yeah, diacylglycerol. And the glycerol is coming from this, which makes sense, because we now know from our biochem that this is a glycerophospholipid. And so now what happens if you cut it in half? Well, one half has to have the glycerol, diacylglycerol. Cool. That'll help me remember what that last G is better. I hadn't connected those two things, but that's true. And then IP3 is the other half of it, and that's a signaling molecule that goes down to the sarcoplasmic reticulum. All right, so
Which is probably a fatty acid?
yeah,
Okay.
yeah, inositol. Yeah, I don't know the structure of it, but it's part of a fatty acid. So, all right. Practicing the other half in your head really quick. Just what are the two things that come off of PLC's cleavage of that one molecule, and then what's the protein kinase, and what's in the sarcoplasmic reticulum. And then thumbs up when you got it. Okay, awesome. And now we can move on. So make sure that you've got a review on everything. I'm going to go ahead.
So, quick question.
Yeah, what's up? No, this is good.
So calmodulin. Sorry, just say it again.
No worries.
Calmodulin binds the calcium, and it's basically a transport for the calcium.
So
and it goes and acts on the MLK. Because I know there's a few questions that were asked that always come like, okay, is it calmodulin or is it MLCK?
So calmodulin is the one that interacts with the calcium directly, and based on calcium levels, calmodulin will talk to the MLCK, and he didn't go super into detail on why doesn't calcium just tell the MLCK to go do its job. Probably because calmodulin talks to some other things that we're not expected to know, and so it's more complicated. Just know that calmodulin is activated by the calcium, and then when the time is right, calmodulin will tell MLCK, go get a real job. Go and do your job down with with the myosin in smooth muscle.
Okay. And with smooth muscle, because he didn't go into smooth muscle, but how does it Because I know the sarcomeres, like in skeletal muscles, how is that different? I've never seen someone, it's always, the example is always smooth muscle.
Okay. So how is it different than skeletal muscle?
Yeah.
So
Like
it's not organized the same way. So in skeletal muscle, you've got a nice Z disc, you've got actin coming out, you've got the myosin doing its dealy. Looking familiar?
Mm-hmm.
And then you got the actin over here, and then you have titin doing its dealy, and nebulin, and blah, blah, blah. But
smooth muscle
in smooth muscle, it's not organized like this at all. There aren't Z discs. It's this just crisscrossed, it looks sort of, a tangle isn't exactly the right word, but it is not organized in a linear fashion. And so you've got these things just whirling all over the place, and they grab on and pull at odd angles, and that's why no one ever draws it, because it's ugly. And smooth muscle is different. But it does still have myosin, and it does still have, I'm not going to swear that it has actin, actually, but it does have myosin. And it grabs onto things, and I think actin's there. And it pulls, but it's not linear, and so it's not pretty to draw.
So a cell that contracts, does it contract in one direction?
No,
so
it doesn't.
It just kind of squeezes.
Yeah, so depending on exactly how that smooth muscle oriented all of its myosin chains will dictate how it contracts. It doesn't contract like this most of the time. Usually it's more like that. And so it contracts based on how it's arranged in there. And that's why it's a big hairy mess and no one draws it, because it's not consistent either. And that's why smooth muscle is good for like sphincters. You want them to not be a linear contraction. You want it to be like all around. And so those ones, it's probably more circular-ish. And then it probably also has some up and down dynamics just to kind of not only do it this way, but then pinch. Mm-hmm. And so, yeah, smooth muscle's cool.
Okay.
Just not when you're an artist. Or not an artist. All right, so yeah, an MLCK. Quick question. Calmodulin, MLCK, are they in skeletal muscle?
No.
No, they are not. Okay? You don't use calmodulin and MLCK to turn on your myosin heads in skeletal muscle. In skeletal muscle, you have the action potential comes in and it triggers release of calcium, and then that calcium directly talks to the, the tropomyosin, troponin, and all that jazz, and then you have.
So making a comparison,
there's none of that.
Is to smooth muscle as troponin as to skeletal muscle.
Very similar. Troponin and calmodulin. Troponin, skeletal, calmodulin, smooth.
Okay, and then MLCK is to smooth muscle as, what's that? The longer, not the troponin, but the tropomyosin? Because it acts on the tropomyosin to move it from the...
MLCK is kind of just its own unique deal. So it goes and talks. It's the thing that makes so that you're not having a calcium ion going straight down into the smooth muscle. You're sending this instead of the calcium ion. And so that's probably the nearest analogy I could call it, even though they act a little bit differently. But so, and I need to review my smooth muscle on whether or not there's actin and all that stuff in there. But hopefully you know that these two are in smooth muscle. That's the take home. Okay, calmodulin and MLCK are only in smooth muscle. All right, that's it for number 1. I'm going to go ahead and stop this video and then do number 2. So the second G protein.